To bring Cure CMD's scientific director to the stage. Since Dr. Gustavo Dziewczapolski took the helm of Cure CMD's research, he has become an excellent listener to the community and experts, and has provided helpful directions to 4 subtype-specific scientific meetings that precluded this conference in the 5-conference series. Please extend a warm welcome to Dr. Gustavo Dziewczapolski.
I have to use this one. Hey, this is me, surfing in San Diego. But that's not what I want to show you today. Okay. Good afternoon. I'm new in this family. I joined this community only 10 months ago. Previously, I worked for more than 20 years in the lab in the field of neurosciences. I did my original contribution as a scientist in the Alzheimer's and Parkinson's disease. Very well-known diseases and pretty well-funded. When I jumped into Cure CMD, I knew I was coming to a field of rare or even ultra-rare diseases. And I started to attend these conferences thanks to this grant that we had to organize 4 different focus conferences, and talking with the experts and reading the papers. And soon I got fascinated by the resources without too much funding that they have, and all the tools and animal models and ideas especially. And another very great feature is that the willingness to collaborate. That is very important for smaller fields like this. And I also was impressed by the very important role that Cure CMD played to create this movement, this strong community, the families and doctors and researchers. So, according to the program title that I didn't write, I should talk about Cure CMD's research roadmap for each of the 5 CMD subtypes. So if I can imitate the GPS voice, I would say recalculating. Because there are alternative routes to follow for each of the subtypes, and it will be new routes for sure as new discoveries are coming from the research and units. Also, as most of the people think in the field, the best therapies at the end will be combined therapies. For example, combining gene correction with an antifibrotic drug to get rid of the fibrosis and give some space for new muscle fibers to repopulate. And so my idea today is share examples of some of the research that is being conducted for the different subtypes. And at the end, I will show you a scheme of potential therapies that are already in the pipeline from the discoveries in the lab in the path to get to real therapies for patients. And if you want more complete picture of the current research for each specific subtype, you can go to our YouTube channel and look for the webinars, Ask the Experts and Community Update that we did, one before and one after each of the specific reunions that we did for LMNA first, LAMA2, collagen 6, LAMA2, and SEPN1. So, exciting examples of current research. I will start with collagen 6, and there was a group of patients presenting typical clinical symptoms plus typical biopsies suggestive of collagen 6 deficiency, but they were negative. They didn't find mutations by exome or by whole genome sequencing. So this has been done by a big consortium of researchers and doctors from 3 different continents: Europe, America, and Oceania. And they use a technique that is called transcriptome sequencing, or RNA-seq, that instead of reading the message in the DNA, they go down here after the transcription that the first RNA message is formed. There is another regulatory step that there is a splicing where all the introns in the green here are left out, and there is the message of the exons that is going out of the nucleus to be translated to proteins. So the idea of this technique is that they collect the RNA and they can study by the number of transcripts of that message if something is wrong, and if something is wrong, they can go back and check where can be the mutation that probably is hidden in the introns. So they found a deep intronic mutation which causes severe Col6 by adding an unwanted extra piece of protein. So the protein it has the normal sequence, but it adds a little piece of protein that is coded in one of the introns that it shouldn't be there. And this is now the most common single mutation in the collagen 6 spectrum disorder. And the nice thing about this is that there are already projects going on to try to get rid of that mutation by a technique called exon skipping that you have heard, because it's the technique that is used to in this drug that was approved last year for Duchenne. So the idea is that if you have a faulty gene with the intronic mutation that have this extra piece of a protein unwanted, you can inhibit that, do the exon skipping, so you get rid of that piece, and you will get exactly another normal copy. This was always normal, and now you can go back to the normal by skipping that piece that shouldn't be there. So it's a very promising, or looks like the easier gene therapy to try to because you will go back to the normal state, totally normal, with 2 copies perfectly fine. So for LAMA2, an example, this is the laminin 211, and one of the functions is to bind with these 2 proteins, integrin and dystroglycan, that are sitting in the membrane of the muscle and stabilize the muscle. When there are mutations in this, in the alpha chain of the laminin, in this case I'm showing a partial mutation, so this piece is gone, but it could be the whole protein that is gone. The idea behind this experiment is to use a recombinant protein. In this case, they use 2, one here, alpha LNND, and miniagrin, to be able to use one of the isoforms that is not the 211, it's the 411 that is already also in the muscle, but is by itself is not that good at doing the job of the alpha-2. But now with the linkers, it can again bind to this protein and stabilize the membrane, the muscle. And this was published last week in Science Translation and Medicine. It's a journal with very high impact. That means that a lot of people read it and is aware of that. And so the idea is now to move as soon as possible this type of therapy to the clinics. And because they try overexpressing these 2 proteins, these 2 linkers in animal models, and they recover the muscle force and size, increase the overall body weight, and extended the lifespan expected for the wild-type mice. I think it's, the paper says that 5 times compared to the animal with the mutation and no treatment. Alpha-dystroglycanopathies. Did I pronounce it well, Rachel? She's okay. Again, the dystroglycan is a protein sitting in the membrane, and it has a collection of sugars here that are inserted by 18 or 20 different enzymes. So any defect in those enzymes will provoke reduced glycosylation there, a defect in glycosylation, and the binding of the lamin, it will get lost. Not now because the lamin is wrong, but because the binding site in the dystroglycan is wrong. So again, in this area, they are looking for linkers, and there is a company, Genzyme, that is trying Biologic Hinge. It's a kind of antibody that recognizes an epitope in this protein and another epitope in the dystroglycan, so it can recover the binding between these 2 proteins and make it functional. They tested this double protein in animal models also, and they improved the muscle function and protected the muscle membrane from the exercise-induced damage in that animal model. They stress it with exercise, and the animals don't get the ruptures of the membrane as the animals without treatment with that linker. I want to go with a couple more ideas in the field of alpha-dystroglycanopathies because, as you know, it comes most of the times accompanied by cognitive impairment. And of course, the cognitive aspect is difficult to study, very difficult to treat, and difficult to manage. For many families, it's the most detrimental factor affecting the quality of life. And as I say, it's very difficult to study these problems, but there is some groups doing some interesting job, and we have one in San Diego that is doing one of the techniques that we call disease in a dish for drug discovery, that is taking somatic cells like fibroblasts from the skin from a patient with a mutation, and they can reprogram that cell like going back in the clock of the cell. So it's now you convert that cell to a pluripotent stem cell, and then you can expand that cell, so you grow the cells, and then you can differentiate that cell in different types of tissues, muscle, cardiac muscle, and neurons and astrocytes, and all of the other types. So what the group in San Diego is doing, they collect some cells from alpha-dystroglycan patients, and they are differentiating that into neurons, and they notice that it is the self-formation of neuronal networks with synchronized firing. So they are connected in a synchronized way. So when there is certain stimulus, all of them responded at the same time because they are in a network. And it could be a model where to start looking for some answers. Like in the aim of this group, they are trying to screen many drugs because they have a facility to do that, a lot of thousands of small molecules, and try to modulate the synaptic plasticity. That synaptic plasticity is what make the connections between neurons stronger. And if that is happening with one of these molecules, it could be try to modulate the synaptic plasticity in the brain of the patient. And something a little bit more futuristic, but it's also in April this year, there were 2 papers back-to-back in the Nature journal. They created human brain organoids that are, instead of that stem cells in a plate, these are 3D mini brains. And with this new technology, this is a good method to mimic human fetal brain development, and could be used to study neurodevelopment and neuropsychiatric disorders. Long from now, but it's there. Okay, so now a little bit of flavor for an experiment in the LMNA, in the laminopathies. In the laminopathies, usually, I think, close to 100% of the times, the pattern of inheritance is dominant, so requiring only one mutation in only one copy of the gene to produce symptoms. So there is a group in France that we were collaborating with them for numerous years, and they are trying to 3 different approaches. One is gene inhibition therapy, so using a genetic tool to block the expression of the bad gene. So they will have a situation with only one copy that is good, and the one that is not good will not be there. But of course, the question is if the 50% of the good protein will be enough to recover the phenotype. For the some results that they have in mice, it looks like it will be very difficult. So the other approach will be gene augmentation therapy. So if we have, again, the same cell with a faulty gene, they will introduce a functional gene, a new gene, wild type. And so you will have this situation with two copies of the genes that are okay, and you still will have the bad copy. And but with that increase in the good copies, it should be a balance toward producing functional protein more than the unfunctional protein. And the third approach that it looks like it would be the best, but of course it requires more technical complications, is to do both, use this tool to block the bad gene and use the other tool to introduce a new copy of the gene, the correct gene. And last but not least, the potential of new therapies, new therapeutic targets for SEPN1. I'm using one of the movies that was made by Besat Mosaget. Yeah, that. Dziewczapolski, yeah. So I will show you, I will play the movie if I can. Yeah. So we have Alan Bex here to correct me if I'm saying something that is not right. It was known for a long time that these two blue structures here that are part of the sarcoplasmic reticulum, the SEPN1 is sitting there. And for some time, they knew that the SEPN1 was involved in some of the oxidoreductase activities of the cell, and also that they were involved in some regulation of the calcium molecules. I will try to stop it. So I can show that the calcium, no more batteries. Okay, the calcium is inside the reticulum sarcoplasmic, and when there is a signal for the muscle to contract, the calcium will go out of the sarcoplasmic reticulum through the RYR channel and go to the sarcomere and make the myosin and actin interact and contract. And then, of course, you need to have the calcium going back to the storage for the muscle to relax, and that has to be very well controlled. And there is that other protein there in red, ARO1, that it looks like ARO1 oxidized the SERCA channel, that is the channel where the calcium is going back in, and the SEPN1 does the opposite effect of reducing that. So that balance has to be very good. So now, knowing this scheme, there is, of course, more targets and more possibilities to play with that system to find out many of the answers that there is still don't know in the field. And I want to share with you the chart that we were putting together that we call the pipeline of potential therapies, that we will go through some edits, but we want to upload it to our website so people can go out there and see where the therapies are. They are divided by subtype in the left, that and the first column in yellow, the second column after the subtype is the therapeutic agent or intervention, and how it will move with these arrows through the preclinical research phase, in vitro and in vivo, and then if it moves to clinical research, clinical studies or clinical trials, to go through regulatory approval, and finally to patients. So we have listed all the therapies that are out there. That I think there are a couple that I couldn't find out yet very well where they go. I have to read some more papers. And there are some that are common to all of them. That is up there, the patient registry, that is very, as the previous talk, very important for us to know number of patients, and that's always useful when you approach a company to convince them or talk to them about doing a therapy program in these diseases. And at the bottom we have, I put the hyperinsufflation study that also goes across. The study was done with the LAMA2 and collagen 6 patients, but SEPN1 and the other subtypes that have complications, respiratory complications, will be there too. And of course, the last one is the biomarkers and outcome measures that many labs are working in finding those instruments to be able to organize a clinical trial and have a good outcome measure to demonstrate if the therapy is going well or not. I will respond the first question without you asking. Yes, you can help us. We need donations and fundraising from you. It's fundamental that we inject funds to move the pipeline towards the real and approved and effective therapies for you and for patients and families. So thank you, and I can take from the second question up.